Prof. Hegar, all attendees. Also, good morning for Professor Rensen as well. Would like to welcome you here in Indonesia, and hopefully we will be able to see you directly in Jakarta, not really via this online webinar. Attendees. Also, good morning for Professor Rensen as well. Sorry. Yeah. Yeah, in this afternoon, we would like to learn about the role of brown adipose tissue in cardiometabolic health. So from the cluster of metabolic, vascular, and aging from the IMERI, we would like to invite you, Professor Rensen, to share your thought, your experience on the role of adipose brown tissue. And we're really very happy to have you here. And before we begin our session, I would like to invite the head of the Indonesian Medical Education and Research Institute, Professor Hegar, to give his opening speech. Professor Hegar, please, time is yours.
Okay, Dr. Diki, thank you very much. Good afternoon and also good morning, I think, for Leiden, yeah, Professor Patrick Rensen. The Honorable, our guest expert, Prof.dr. Patrick Rensen, our Head of Metabolic Disorder, Cardiovascular and Aging Research Center, IMERI, Dr. Diki Tahapari, PhD, Specialist Internal Medicine, Consultant in Metabolic, Endocrine, and Diabetics as well. Professors, seniors, and also all researchers and educators. First of all, thank you very much for coming to the scientific afternoon lectures. When I read the topic that will be discussed this afternoon, a greater curiosity arises on this topic. What is the function? And also, what is the importance as well? Because we know that a brown adipose tissue together with white adipose tissue makes up the adipose organ. And also blood adipose tissue is reported abundant in newborns. And it is also represent and metabolically active in adult, but its number decreases as human age. They are good for us in normal amounts. But what happens when we having too much in our blood? Will it give something wrong with our cardiometabolic health? So I think we are so lucky today that we can attend to this amazing topic this afternoon. And I'm sure that there will be numbers of interesting issues to be discussed, such as the functions, metabolic, how to lose, and also other important issues as well. So once again, thank you very much to Prof. Patrick Rensen for spending your time to share your knowledge and experience with us this afternoon. And I also hope that IMERI, Faculty of Medicine, Universitas Indonesia, especially Metabolic Disorder, Cardiovascular, and Aging Research Cluster, and Research Laboratory, Department Medicine, Division Endocrinology, Leiden University Medical Center, will further develop various tests of convergent research as our efforts to create better quality of healthcare and also quality of life. And of course, also building and sustaining Faculty Medicine University Indonesia and Leiden University Medical Center education and research capacity. Okay, thank you very much. Please enjoy the lecture. Thank you, Dr. Diki.
Yeah, thank you, Professor Hegar, for the opening speech, and thank you for the support of the IMERI to this guest lecture on future collaborations. Before we begin our session, I would like to introduce Professor Dr. Patrick Sian Rensen. He is currently the Professor of Metabolic Aspect of Vascular Disease, Leiden University Medical Center, Leiden, the Netherlands. He is also the guest professor at Xi'an Jiaotong University, Xi'an, China. He also established the investigator of the Dutch Heart Foundations, and also is currently the chairman of the European Lipoprotein Club. And his research group studies the role of lipid and glucose metabolism in cardiometabolic diseases, with the main focus on brown adipose tissue activations as a strategy to combat obesity, type 2 diabetes, and atherosclerotic cardiovascular disease. Okay, please, time is yours, Professor Rensen.
Thank you very much, Dr. Tapahari and Professor Hegar, for inviting me. It is really, really nice pleasure for me to speak to this audience. I must say it's the first time I speak to an Indonesian research community, although it's via Zoom. But I really, really hope that in the future I'll be able to come to Jakarta and share more thoughts than in this approximately half an hour. So yeah, I'm working at the Leiden University, but also I'm trying to set up a metabolic research facility in Xi'an in China, because one of my former PhD students, Yanan Wang, is now a professor in Xi'an, and she's developing her lab there. So I have been interested for a long time in lipoproteins. So you know that lipoproteins are the particles that transport the hydrophobic lipids through our blood, which are the cholesterol, esters, and the triglycerides in the core of particles that are surrounded by a monolayer of phospholipid, cholesterol, and apolipoproteins that are involved in the synthesis, the catabolism, but also the uptake of lipoproteins from the circulation. And just to refresh your memory, I guess many of you know this already, but the small intestine takes up cholesterol, esters, and triglycerides. They are packed in chylomicrons, and these are mainly rich in triglycerides, and they deliver fatty acids towards metabolically active tissues like the heart and the muscle and the white fat. And they do so by liberation by lipoprotein lipase. That is an enzyme that liberates fatty acids from triglycerides. Those are taken up by the heart and muscle for beta oxidation and ATP production. And the majority after the meal is taken up by the white fat for storage as triglycerides. So by this process, chylomicrons are enriched with ApoE, and the remnants are taken up mainly by LDL receptors in the liver. And the liver itself is also produce lipoproteins in the form of very low density lipoproteins. And these have essentially the same function. They also deliver fatty acids towards this tissue, but mainly under fasted condition to the heart and the muscle, for example, during the sleep, where the heart is provided with fatty acids, so the glucose is still able to feed onto the brain. The VLDL particles also become enriched with ApoE and also are taken up as remnants by the liver, and full lipolysis of VLDL gives rise to low density lipoproteins that's mainly cholesterol rich, and that serves to transport cholesterol to other tissues, for example, membrane synthesis, but also for hormone synthesis. Like the adrenals, they depend on cholesterol from the circulation to produce our stress hormones, like glucocorticoids. And also cholesterol is a precursor for vitamin D, of course. So this is all in a good situation, but in this society of metabolic overload, we are often in a positive energy balance. So we consume more energy, we take in more energy than we expand, leading to expansion of the white fat, and that in the end leads to overflow of lipids towards the liver. So hepatocytes become lipid-filled. That leads to insulin resistance and non-alcoholic fatty liver disease. But also there is a lipid overflow towards muscle and heart cells, like myocytes, cardiomyocytes, and also there that leads to insulin resistance. So that not only leads to an increase in glucose in circulation, but also in lipids, because the liver starts to produce a lot of VLDL particles, and the heart and the muscle do not take up as much fatty acids. So it leads to an increase in triglycerides and LDL in circulation, together with a reduction in HDL. And in the end, the increase in atherogenic lipoproteins in circulation will lead to an increased uptake of cholesterol by the arterial wall, and that starts the atherosclerotic process. HDL is there, a good lipoprotein that is thought to extract the cholesterol again from the aortic wall. So in my group, we are interested in all these aspects of lipoprotein and glucose metabolism, and we are looking at the role of the gut, but also of skeletal muscle, of the brain and the liver in handling all these overload of fat and glucose. And these are the postdocs that work in my group, and they are supervising approximately 15 PhD students, helped with 5 research technicians. And today, I'd mainly like to present to you some data we obtained on brown fat activation, both in small mammals, but also in humans. So you probably all know that brown fat in neonates is located between the shoulder blades as quite a large fat pad. And the function is quite different from white fat, because white fat cells are very large cells and they're mainly there to store energy, while brown fat cells are smaller. They contain a lot of small lipid droplets, but also mitochondria, and these mitochondria serve to oxidize lipids. And normally when lipids and glucose is oxidized, that leads via the respiratory chain to a proton gradient across the inner membrane of the mitochondria. And normally this proton gradient is used in muscle tissue and heart tissue to generate ATP via ATP synthase. On the other hand, in brown fat, there is a very specific molecule called the uncoupling protein 1, and it forms a sort of a pore within the inner membrane of a mitochondrion, and that leads to a flow back of protons to the matrix of the mitochondrion. And that is not coupled to ATP production, but to heat production. And the heat that is being produced is then circulated via the aorta in the whole body to keep the baby warm. We always thought, as Professor Hegar already told us, that brown fat declines with age, and indeed we lose the fat pad between our shoulder blades. But it wasn't until about 2009 where people were deliberately exposed to cold by cooling between two water-cooled blankets. And after that, we gave them an 18F fluorodeoxyglucose, which is a glucose analog. And when you then make a PET CT scan, then you see quite a lot of uptake of glucose by regions above the claviculae, but also around the aorta. And those regions appeared to be UCP1 positive. So we know that here we have brown fat in human in locations that are shown here, but also we know that white adipose tissue is able to brown because brown fat cells can develop between the white fat cells in our white adipose tissue depots. So how does it work? So when our environmental temperature decreases, that is sensed by TRP channels in our skin. And you know that TRP channels were discovered a long time ago, but the discoverers of TRP channels have been awarded the Nobel Prize this year. But there are channels that send cold, leads to an afferent signal that is perceived by the temperature control center in the hypothalamus. And when that center senses a cold environment, there is a sympathetic outflow towards brown adipocytes, mediated by noradrenaline secretion that binds to beta receptors on brown adipocytes, and that leads to a thermogenic program there, and that is coupled to the uptake of glucose. But we also know that the brown adipose tissue is able to take up triglycerides. And that comes from a very nice study from a group in Hamburg, Jörg Heer and Alexander Bartold. They showed that mice that are hypertriglyceridemic, when they expose these mice to cold, by putting them in a fridge at 7 degrees, you see a very rapid decrease in triglycerides. And basically the turbidity in the plasma is completely cleared by a few hours of cold exposure. So we tried to see what is the mechanism why the brown fat takes up or is able to clear the blood from triglycerides. And for doing that, we made use of recombinant lipoproteins that I developed a really long time ago when I was still a PhD student here in Leiden. So what I did was I made lipoproteins from commercially available lipids, and we were able to trace these lipoproteins with cholesterol trioleate, which is a triglyceride where the tritium is bound in the fatty acids of the triglycerides. But we can also label these particles with cholesteryl ester, 14C cholesterol oleate. We can inject these particles into the vein of a mouse and then take out after 15 minutes several organs to see where the fatty acids and also the cholesterol oleate ends up. So Geert Hoeke was a PhD student in our lab, and she did it in mice, showing that that already at room temperature, quite a lot of fatty acids are taken up in the brown fat, and it was quite enhanced when we expose these mice for a few hours to 7 degrees. And you really see quite a high uptake of the fatty acids by the brown fat. On the other hand, the cholesterol esters are also increased in uptake by the brown fat, but you see here that brown fat takes up 60% per gram of fatty acids and only 6% of per gram of cholesterol esters, showing that there is a selective delipidation of the fatty acids from lipoproteins by the brown adipose tissue. And these and other studies have led to this mechanistic figure that when the environmental temperature is decreased, there's noradrenaline being released from nerve endings in the brown fat, binding to beta receptors that leads to the production of cyclic AMP in the brown fat cell, activating protein kinase A, that leads to phosphorylation of intracellular lipid hydrolases, and then fatty acids are liberated from lipid droplets to feed onto the mitochondria. So the lipid droplets are depleted, and they are repleted again by the uptake of fatty acids from the circulation, and these are derived from triglyceride-rich lipoproteins that become then remnants, and these remnants are taken up by the liver. Also, we know that glucose, of course, is taken in. We don't think that glucose is directly burned by the brown fat, but it is used for de novo lipogenesis and lead to more fatty acids that are incorporated again in lipid droplets before noradrenergic signal leads to the liberation of them for again lipolysis and oxidation. So from all of these studies, we were able to add brown fat to the textbook as one of the metabolic organs that take up fatty acids by lipoprotein lipase induced liberation of fatty acids from triglycerides. But in the white fat, the fatty acids are used for heat production instead of ATP production and for storage. So since we know that many groups have looked at the effects of brown fat activation on adiposity and non-alcoholic fatty liver disease, I won't talk a lot about this, but we were mainly interested to see if brown fat activation can also improve dyslipidemia and atherosclerosis. And for that, we made use of mouse studies. And of course, you know that a mouse mainly carries the cholesterol as HDL cholesterol, while a human mainly carries cholesterol as LDL cholesterol. So in Leiden, we made a very specific mouse model. That is the ApoE3 Leiden CGP mouse model. And this mouse model is quite humanized. It behaves like humans, because now most of the cholesterol is indeed in the LDL and the VLDL. And what is very good about this model is they respond in a human-like manner to lipid modulating strategies. And it is quite unlike the ApoE and LDL receptor knockout mice, because many of lipid-lowering interventions, like with statins, for example, they depend on the uptake of triglyceride-rich lipoprotein remnants via ApoE by the LDL receptor. And of course, when a mouse doesn't have ApoE or doesn't have the LDL receptor, a statin is not able to reduce lipids from the plasma. So this is quite a good feature of our model, and that's why we also collaborate quite a lot with pharmaceutical industry to see if experimental lipid-lowering drugs also attenuate atherosclerosis. So Jimmy Berbet, who is a late assistant professor in my lab, used these mice, and he fed them first a cholesterol-rich diet to increase the cholesterol levels in the plasma. And then he provided the mice 3 times a week with a beta-3 receptor agonist that sort of mimics the sympathetic outflow in towards the brown fat. And then after 10 weeks, we took out the heart to look at the atherosclerosis progression. But first, we house the mice in metabolic cages where we can measure energy expenditure. And after a shot of a beta-3 receptor agonist, we see an increase in energy expenditure. And also the overall energy expenditure is increased. And it's mainly caused by an increase in fat oxidation. As I explained already, brown fat takes up fat and glucose to increase fat oxidation. So that was linked to a reduction in the fat pads. But when we look at the fat pads, we also see that these become positive for UCP1. And that shows that if you are exposed to a beta-3 receptor agonist as a mouse, then the white fat starts to brown, and there are brown fat cells appearing within the white fat cells. And again, when we inject our particles into these mice, we see that different brown fat depots take up quite a lot of fatty acids, and the remnant of the particles is taken up by the liver to an increased extent. And that leads to an overall decrease in triglycerides in the circulation, but also a decrease in the cholesteryl esters in the circulation. And that is causal to less atherosclerosis progression. You see here in the aortic valve route, you see lower atherosclerosis, but also at the various distances from the aortic valves, you see that the atherosclerosis is decreased. And this is basically the mechanism why we think it's all happening, because when we activate in the mouse the beta-3 receptor, that leads to a high increase LPL-dependent fatty acid uptake, and that is coupled to a very efficient uptake of the remnants via the ApoE LDL receptor pathway by the liver. So the cholesterol in the plasma is lower, so there is less pressure on the aortic wall, and there's less atherosclerosis formation. So later on, we realized that also the excess of surface from these particles is liberated in circulation to bind to lipid-poor ApoE1. So small HDL particles are being formed, and they can pick up cholesterol from the arterial wall and deliver the cholesterol esters via the SRB1 to the liver. And all the cholesterol that is provided to the liver via remnant particles and via LDL can then for some part be excreted into the feces as bile acids. So we have shown that the ApoE and the LDL receptor are both critical. So we thought that if we would increase the LDL receptor on the liver, we may sort of add to the anti-atherogenic effects of brown fat activation. And indeed, Geert de Hoeke and Chen Zhu have shown in our lab that if we treat these mice also with a statin or a PCSK9 inhibitor, then that leads to an additive effect on atherosclerosis. But also if we prevent the enterohepatic circulation of bile acids by giving these mice bile acid sequestrant, so that leaves all the bile acids that are produced in the bile to be secreted from the circulation from the mouse into the feces, then that also adds to the protective effects of brown fat activation on atherosclerosis. So we are looking also in the lab for new therapeutic strategies apart from beta receptor activation, and we are very interested in, for example, in FGF21, which is a sort of stress hormone that is produced by both brown fat and the liver. And you may know that there are clinical trials now being done with long circulating FGF21 analogs, and they have been shown in humans already to increase insulin sensitivity, but also to reduce atherogenic lipids. And Thong Liu in our lab has provided also these long-acting FGF21 molecules to mice. And we show really a very high activation of brown fat, but also a browning of white fat, leading to increase in triglyceride turnover and leading to, again, the remnant being taken up by the liver, leading to lower cholesterol in circulation and the prevention of atherosclerosis formation. So we think also in humans, FGF21 may also increase turnover of lipoproteins to lower atherosclerosis, but of course that is what we should see. What I also really like is that we recently, together with a group in Copenhagen here in Europe, have shown that if we cold expose mice and we look at all the G-coupled protein receptors, G-protein coupled receptors, we saw that there was an orphan receptor, GPR3, that was very much increased. And when we overexpressed the GPR3 in the brown fat of mice, they were protected against diet-induced obesity. And with our recombinant lipoproteins, we were able to see a very high increase in the uptake of fatty acids by the brown fats, both in a constitutive and an inducible GPR3 overexpressing mice. And so in the end, by doing a lot of different studies, we've seen that lipolytic signals that are produced by cold exposure, they are inducing GPR3 expression in the nucleus, leading to GPR3 on the cell membrane that without an exogenous ligand can activate the intercellular thermogenic program to reduce obesity in this context. And we are now also doing studies with GPR3 overexpression in brown fat in mice by lentiviral overexpression. And we have just performed that in our APC and CTP mice. So soon we will see if this also improves lipoprotein metabolism and also reduces atherosclerosis. So, of course, I want to talk about the last 5 to 10 minutes about the effects of brown fat in humans. And these studies in our lab are being supervised by Mariette Boon and a Spanish postdoc, Borja Martinez, and they're conducted by our PhD students. And we were inspired, of course, by the recent studies that were published in Nature Medicine. There was a group in America. They looked at almost 140,000 PET-CT scans that were basically made for tumor detection. And they were also made just at room temperature. And they saw that about 6% of these scans, they showed active brown fat. And when we looked at the persons that did show active brown fat, there was less prevalence of type 2 diabetes, less prevalence of coronary artery disease, and in both people who were obese or had overweight or had a normal body weight. And also the people who had active brown fat had less triglycerides and higher HDL cholesterol. But of course, this is completely association and we don't know any causation here. So we have done now some studies where we exposed, this is a study from Maastricht in Netherlands, where people were exposed to a low temperature in metabolic rooms for 6 hours per day during 10 days. And you'll see that that does increase the brown fat activity measured by a PET-CT scan again in both males and females. And a Japanese study have shown that if people were cold exposed for only 2 hours a day at 17 degrees for 6 weeks, then they lose some body fat, which is not much, only about 700 grams in 6 weeks, but that may be sufficient already to reduce the ectopic fat. And also the cold-induced thermogenesis was increased after 6 weeks of cold exposure. Our group, our collaborators in Maastricht have shown that also when you cold expose type 2 diabetic patients for only 10 days at 16 degrees, then the uptake of glucose by the brown fat is increased. But a very nice observation was that also the glucose infusion rate was increased during a hyperinsulinic euglycemic clamp, which shows that these patients become much more glucose tolerant. And my favorite hypothesis is that their ectopic fat reduces by only a few days of cold exposure, and it leads to a higher insulin-dependent glucose uptake of those tissues. And this is a study that was published quite recently from Scandinavian, where they looked at winter swimmers compared to people who did not winter swim. So basically, they dipped themselves in ice cold water twice a week in Scandinavia, followed by a hot sauna. And they were able to see after winter that they showed much higher cold-induced thermogenesis, meaning that that winter swimming is at least associated with an increase in brown fat activity. So with respect to lipoprotein metabolism, we have cold exposed males and females of about 20 to 25 years old, so students here in our facility, and we looked at the lipoproteins. But we did observe an increase in angiopoietin-like 4 in the circulation, together with an increase in fatty acids, also an increase in triglycerides, and an increase in HDL cholesterol in these persons. And that is completely consistent with what we also know in mice, that white fat starts to produce angiopoietin-like protein 4 to liberate fatty acids in the circulation. So these are going to the liver, they drive the VLDL triglyceride production. That's what we see here. And that stimulates the uptake of fatty acids by the brown fat, leading to an increase in HDL cholesterol. But of course, in mice, we mainly see that the total triglycerides are reduced by the initial, we see an initial increase in total triglycerides. So we wanted to know if we not only have a production of triglycerides, but also, of course, and consumption of triglycerides. So we performed lipidomics on plasma that was taken between 0 and 120 minutes after cold exposure to the shivering temperature of young men and women. And then you see for the smaller triglycerides and the more saturated triglycerides, we see a decrease in these species over time, because the more blue, the more decrease there is. And on the other hand, for the larger triglycerides that are more unsaturated, we see an increase in their species. So we think that the circulating triglycerides are rapidly being hydrolyzed, leading to a decrease, but the liver starts producing more unsaturated, larger triglycerides. And as a compensatory mechanism, because the liver is also sympathetically innervated or activated after cold exposure. And we are now doing some studies in mice with an inhibitor of the lipolysis in white adipose tissue. And we do see that a similar effect in mice also on the production of very large triglycerides by the liver that's completely abrogated when you feed this mice with HDL statin, which prevents the hydrolysis of triglycerides in the white fat. So of course, in humans, we look at drugs to activate the brown fat, and Aaron Sykes already in 2015 showed that mirabegron, a beta-3 receptor agonist, is able to activate brown fat. It increases the metabolic rate and the FDG uptake, but it was related to an increase in heart rate and systolic blood pressure. And you should realize that the 200 milligram that he used is quite higher than the 50 milligram dose that is normally used to treat a hyperactive bladder. And so together with a group in Vancouver, we gave now persons the pharmacological dose of 50 milligram, and we did not see brown fat activation, basically. And a PhD student in our lab, Eileen Kuipers, she used differentiated, immortalized, and cloned human brown adipocytes to show that mirabegron does lead to brown fat activation, because there is an increase in liberation of fatty acids and glycerol, but this effect was completely abrogated by a beta-1/2 antagonist. And also noradrenaline is activating the fatty acid and glycerol release, but also completely prevented by a beta-1/2 antagonist, meaning that the effects of noradrenaline and mirabegron in human brown fat is probably mediated by the beta-2 receptor agonist. And that's what we published together. So in a mouse, the brown fat is mainly activated by a beta-3 receptor agonist. And in humans, probably also in an in vivo situation, the beta-2 is causing the activation of the brown fats. So that's what we currently trying to see in humans. We gave them an intravenous injection of salbutamol as quite a selective beta-2 receptor agonist, with and without beta-1/2 antagonist propranolol. And our preliminary data really show that salbutamol gives quite a high increase in brown fat activation, and much higher than even mirabegron is able to do. So I wasn't able now today to talk about many other therapeutic strategies that may also be important in the end. Today I talked about the beta-3 receptor in mouse, the beta-2 receptor in humans, and FGF21 and GRP3. But we also showed that other GPR receptors like the cannabinoid-1 receptor and GPR120 are involved in mouse brown fat activation, and probably that can be extended to also human studies. We have seen also that microbiota and short-chain fatty acids are able to activate the vagus nerve to indirectly activate the brown fat. And we have seen in the recent studies that the brown fat is also under circadian control because of the daily light exposure. And that, I didn't talk about today, but the brown fat seems to be active mainly at the start of the day and much less active later on the day. And so we use that now for chronotherapeutic approaches in mice and in humans. So I hope what you take from this lecture is that brown fat plays a role in the metabolism of glucose, but also triglyceride-rich lipoproteins, that mice can be instrumental to demonstrate that brown fat activation improves many metabolic features, including adiposity, dyslipidemia, atherosclerosis, and also non-alcoholic fatty liver disease. And that mouse studies have revealed numerous pharmacological targets, including the beta-3 receptor, FGF21, GPR3, and also others, including microbiota. And in humans, the brown fat is primarily activated by cold exposure via the beta-2 receptor, we think, but also hormones like GLP-1 and secretin have recently shown to activate brown fat. But we still await long-term intervention studies addressing the potential effect of brown fat activation on atherosclerotic cardiovascular disease. And we just received quite a large grant from the Dutch Heart Foundation, where we are going to cold expose humans for a very long time and look at cardiovascular risk factors and see whether those are improved. So these studies have been performed in close collaboration with many groups at LUMC in the Netherlands, and so far in many countries, apart from Indonesia, and possibly we can collaborate in the near future also on this topic with you. And I really like to thank all my Batmen and Batwomen who really performed all our studies, and basically the PhD students and the research technicians did a lot of the work. And I'd like to really thank them for all their support. And I really like to thank you also for your kind attendance of this lecture. And I would be very happy to answer questions that you may have. Thank you very much.
Okay, thank you very much, Professor Rensen, for a very insightful and really interesting lecture on the role of brown adipose tissue. It's really interesting to see how the progress of the research on brown adipose tissue. We really look to work with you maybe in the future to work on this brown adipose tissue. In relation with the cardiometabolic health, here in Indonesia, we have a huge problem of cardiometabolic health. For example, obesity. It's already about 1/3 of our population. Diabetes about 10%. So the role of brown adipose tissue is really interesting things to study, and could potentially also lead to a new interventions in the future. I'll see a raised hand from Dr. Farid Kurniawan. Please, Dr. Farid Kurniawan, you can ask your question directly to Professor Rensen.
Yeah. Thank you so much, Professor Rensen, for a very interesting lecture. I have two questions. One is regarding the physical activity and diet. You said that it could also, the microbiota could also change this brown adipose tissue. Is there any significant role from the physical activity or certain diet? That's my first question. And the second question is regarding ethnic or race, because I assume that some people living in subtropical countries or tropical countries might have different brown adipose tissue composition or distribution. Maybe you have the data. And what is the effect of intervention, whether if given to this two type of ethnic or populations, whether it will be different or not? That's my questions. Thank you.
Thank you very much. These are really very relevant questions. And yeah, what we know about exercise. I mean, there were mouse studies formed in the lab of Professor Spiegelman showing that exercise releases irisin from muscle cells, and irisin can feed onto the brown fat to activate it. So we performed a large study with exercise together with people in Granada in Spain. But in the end, we saw that if you give them several densities of exercise, that we do not really see that the FTG uptake by the brown fat is increased. So at least the glucose uptake by the brown fat does not seem to be increased. On the other hand, we are now developing also a triglyceride tracer for humans, because I'm quite convinced that we should look at the human brown fat with respect to the fatty acid uptake and not so much the glucose uptake. So the verdict may be still out. What we do see with exercise is we see also in that study, when we look at the feces, a huge change in the microbiota. And we have pinpointed one specific bacterium that is a high short-chain fatty acid producer. And when we give that bacterium to our mice, we see that brown fat is activated with respect to the fatty acid uptake. What we also see is the muscular strength of the mice increases a lot. So yeah, we are having a large program now on seeing, on looking at various gut microbes that we get out of our human studies, and we give to our mice after depleting the gut from microbiota, and see whether that is preventive. And so that is quite a recent area that we are studying. That's one. We do see also, there is a lot of ethnic variation, I guess, with respect to brown fat. What we've done so far is we have looked at the brown fat in Europeids versus Dutch South Asians. So people who are from the South Asian continent, like India and surrounding countries. They, in the past they traveled to Suriname, and from Suriname they traveled to The Hague here. And we have seen that their brown fat activity is much lower compared to people who were born in the Netherlands. And so I do think that if you're living in warmer climates, then brown fat is already less activated, and in time over generations that may lead to genetic programming, so that less brown fat activity is needed. At least when we see in these South Asians that 2 generations ago, people traveled to the Netherlands, and even their grandchildren now have less brown fat activity. At the same time, we often see a much higher liver fats and other ectopic fats in South Asians. And what I think is really interesting, that is a graph here, that we looked at the proteome, so all different kind of proteins in the blood of Dutch South Asians versus Dutch, let's say, white people. And the highest difference was there in FGF21. And normally FGF21 is increased in metabolic stress, but here we did see that South Asian people had lower FGF21, although they have higher body fat. So we have the idea that maybe FGF21 may be especially favorable in treating people from South Asian descent, and possibly also other descent. We collaborate in China, of course, and we see basically the same. Also there, the obesity epidemic is increasing. Also, the liver fat is relatively higher already at a lower BMI. And we think that this FGF21 program or intervention may be of benefit, maybe also there.
Oh, very interesting. Thank you so much, Professor Rensen.
Okay, thank you, Professor Rensen, also Dr. Farid. There is another question from Dr. Sri Suryani Wijaya. Professor Patrick, thank you for the very interesting lecture about brown adipose tissue. Do you have any data about how long the exposure to the cold temperature can induce the activity of the brown adipose tissue? And what is the range of the temperature?
That's a very good question again. We have the brown fat will be inactive when we are in a thermoneutral condition, and for humans that is estimated as between 23 and 25 degrees. So in a regular temperature of 20 degrees, for example, the brown fat is already activated. You see that by a small increase in the FTG uptake. I hope we are able, and that is shown here, we have just been able to make an 18F labeled triglyceride. And we really hope that that can be a new golden standard for brown fat activity in human. But what we have seen is that when people are exposed at 17 degrees, we already see a higher FTG uptake. And I think that the lower the temperature you're exposed with, the higher FTG uptake there is. Of course, if the cold exposure is below our shivering temperature, then also there's a lot of glucose and fatty acid uptake by the muscle for shivering, and that of course brings a lot of heat to the body. So I think the lower the better. Personally, in the winter, I have my thermostat at 17 degrees. I feel a little bit of cold at home, but I have the idea that I constantly activate my thermogenic processes there. I do think that the thermogenic trigger is needed, and that if you don't have the trigger, then very quickly you lose the trigger to activate a brown fat. We have a study in Rotterdam where people who had surgeries, perirenal brown fat was taken, and we could correlate the UCP1 expression with the temperature over the last 3 days in the Netherlands. So I have the idea that the temperature in the previous days determines at least the browning of the brown fat, of the white fat. But if you go from here to a warm country, I think within a few days you lose the trigger, and I'm afraid then the brown fat activity is back to baseline.
Okay, thank you, Professor Rensen. I think that should answer the question from Dr. Sri Suryani Wijaya. Thank you, Professor Rensen. There's also a comment from Dr. Leiden. Very insightful lecture. Thank you, Professor Patrick. And then another question from Dr. Beatrice C. Walter. Thank you, Professor Rensen, for the wonderful presentation about the brown adipose tissue. I want to ask you about the role of vitamin D, to brown adipose tissue. Does taking supplementation of vitamin D has some amplification to the brown adipose tissue?
Oh, that is a very good question. And I'm afraid I don't know the answer by heart. I know the vitamin D receptor is present. We can see it on the RNA sequencing of the brown fat. We do see it there. I must admit that I did not look up in literature whether that has been performed already. I can still do that and send my findings to Dr. Diabetic, but I'm afraid I can't tell you at this moment. Sorry.
Okay, thank you, Professor Rensen. So while we were waiting for another question, I'm really puzzled about this browning, because if you can initiate the increase of brown adipose tissue, like you have like agonists, you say like there's several intervention, right? I think the similar issue with Dr. Farid, whether it will give like a similar response, for example, for those who live in a cold area compared to those live in tropical area. Do you have any thought on that, Professor Rensen, whether it would give similar response? Because of course, in those who live in the cold area, there is a more brown adipose tissue compared to we live in Jakarta, for example, the average temperature is already 30 to 35 every day. Thank you.
Yeah, that's a very good question. I'm quite convinced that if you live in the area like in Jakarta with such a high temperature, then the brown fat cannot be activated by itself, not by the environmental temperature. And I think, then you really should need maybe a beta-adrenergic signal, or maybe one of the drugs that we try to develop, for example, based on FGF21 or based on TPR3, to try and activate the brown fat because at those temperatures there will not be much brown fat activation, and I'm quite sure that there will also be only little browning, if any. So that then we need to do that I'm afraid via pharmacological approaches. Possibly we see that dietary intervention can work. We do see that basically because I'm really think that increasing the fiber intake is very important here. We have done studies first in mice to give mice the digestion product of fiber, which is the short chain fatty acid like butyrate. And if you give butyrate orally to mice, we see a high increase in the sympathetic activation of the brown fat. We also see that satiety is induced. And of course, butyrate is just a fermentation product of fiber. So I have the idea that if we change our dietary intake away from the ingestion of meat, more into fiber-rich materials like chickpeas or like peanuts or like any vegetable that is rich in fiber, then we can maybe via temperature-related stimulus also try to activate the brown fat, but also reduce the food intake, and which is, of course, at least as important to combat cardiometabolic disease.
Okay, thank you. Thank you, Professor Rensen. There's also another question, but I think this is the same thing about my previous questions about if you stay in hot climate, about the activity of brown adipose tissue. I think we already answered these questions. So the next question is about, maybe it will be the last question because there are 2 minutes left. About you show us a slide that there is a substance that can also induce the brown adipose tissue activation, and then it could greatly reduce the LDL cholesterol as well as the atherosclerosis and then you combine with statin and also PCSK9 inhibitor. How much superior is the activation of the brown adipose tissue compared to the use statin or PCSK9? Because the PCSK9 is very powerful LDL reductions.
Yeah, well, that's of course very much dependent on the dose. I mean, if you take the full active dose of the PCSK9 inhibitor, we show in our mice, it is very, very effective. I mean, you can decrease the cholesterol in the plasma by let's say 90%, and it will completely prevent the atherosclerotic process. And that's the same with a statin. I mean, if we just increase the dose of a statin, then we can do the same. What we do in these kind of studies to see if we have an additive effect of two interventions or maybe a synergistic effect, we use a dose of a statin and a PCSK9 inhibitor that reduces the cholesterol in the plasma by only about 25 to 30%, and then see if another intervention, which can be an anti-inflammatory intervention, or in this case, a brown fat activating intervention, and see whether when we then have an additive or synergistic effect. And in the doses that we used, we saw that the brown fat activation had about a similar effect on cholesterol and on atherosclerosis as the statin or the PCSK9 inhibitor, but their combined presence had an additive effect. They were also not synergistic because basically you intervene within the same pathways, like the increase in the lipid turnover of the triglyceride-rich lipoproteins. Okay, I think that in humans, I mean, of course, we have less brown fat in the mouse. So the relative effects of brown fat in the human will always be lower than the effect that you can reach with a statin or a PCSK9 inhibitor.
Okay, thank you. Maybe one more last question from Dr. Farid about following your last answer on microbiota, physical activity, and brown adipose tissue. Do you know which population of microbiota that really affect the activation of brown adipose tissue? Is there any specific composition or bacteria? Yeah, I'm afraid I cannot disclose the exact microbiota because we are now involved in patent protections, but at least I can disclose that it's really the short chain fatty acid producing bacteria that do the trick. And that is basically the bacteria that you highly increase when you change a diet from, for example, a meat containing choline-rich diet to a fiber-rich diet. Then you really increase the microbiota that produces short-chain fatty acids. And I think that is the most important for increasing brown fat activity, but also for reducing total food intake. Okay, maybe I would like to ask one final question as the closing thought of your lecture. Do you think that the activation of brown adipose tissue, what I meant is intervention to activate the brown adipose tissue would take the center stage in the next few years to also control the cardiometabolic diseases?
Sorry, what's the exact question?
Do you think that the brown adipose tissue activation would take the center stage in the management of cardiovascular diseases in the future?
Well, center stage is, I think, for really big words, because I mean, we have just the statins and other drugs that are just too powerful. But I think in the end, what we learned from this research, that we should modify our natural behavior also. I mean, we should modify our eating behavior to lower food intake, to have more vegetable intake, and that will increase brown fat activity. We should exercise more. That may indirectly prevent or activate brown fat activity, but also prevent ectopic lipid deposition. What I do think is that now we know that the beta-2 receptor is basically the receptor involved in brown fat activation, and then we can selectively target that to the brown fats by now knowing which specific receptors are residing on brown fat. That may be additive. I'm not really sure if it will be the center stage of metabolic of combating metabolic disease.
Okay, thank you very much, Professor Rensen, for your sharing your experience. Very insightful for all of us, and we also hear quite a lot of questions. Hopefully, we can meet later on just to discuss for possible collaboration, as well as maybe after the COVID pandemic has gone, maybe you can also come and visit us at IMERI. Before we close this session, I would like to ask for maybe a group photo, if you don't mind, Professor Rensen.
Of course, yeah.
Shall
I stop sharing then?
Yeah, that would be great.
Yeah.
Okay. So I would like to invite all the participants to turn on their camera. And maybe Mbak Tika mungkin dibuat semuanya, Mbak Tika, sama-sama kita fotonya.
Nah, kok diakhiri? Astagfirullahaladzim.
Oke, mungkin silakan Bapak, Ibu dokter sekalian, researcher semua, silakan kalau mau berkenan nyalakan kameranya, kita akan ambil grup foto. Baik, Mbak Tika, silakan Mbak Tika, minta tolong diaba-abain. Okay, maybe can take a picture. We have 2 pages, so maybe the first pages, 1, 2, and 3, and then the next page. One, 2, and 3. Okay, I think that's all for now. Again, thank you very much, Professor Rensen, for sharing a really excellent talk on this session. Prof. Hegar, would you like to say something?
Okay, thank you very much again, Patrick. See you later in the next collaboration research as well.
Certainly. Thank you very much. Goodbye.
Thank you, Professor Rensen. Thank you.
Goodbye.
We also want to just to inform you that we will also
The lift, ya.
Ya, makasih Prof. Makasih Prof. Ya, ini kita ada acara lagi untuk sekitar bulan Februari. Topiknya akan membahas mengenai microbiome in health and disease. Kalau dokter kan peneliti sekalian tertarik, nanti informasinya akan menyusul. Kita akan undang beberapa pembicara dari luar dan juga dari Indonesia untuk membahas peran mikrobiota di beberapa penyakit yang ada, terutama penyakit metabolik. Baik, terima kasih untuk perhatian rekan-rekan dokter, Bapak Ibu sekalian. Terima kasih sekali lagi, sampai bertemu di acara kelas Remba berikutnya. Terima kasih.
Terima kasih.
Terima kasih, Dok.
Terima kasih.